another express epitope is there.
Yes, yes, that is why the express epitope is there. Thanks, Obed. I have turned on the recording now.
Sir,
Yeah, Chandav.
sir, yes, sir. So in the last lecture, like you had told that, I mean, if there is a lack of in the vector also, and what could be the use of that lack of opron? I mean, the uh,
Mm-hmm.
so like there could be multiple level of I mean, oh, what is the word I forgot?
Like
Mul
stringency? Stringency? Are you talking about stringency?
control, multiple level of control.
Uh-huh.
So if there are lack operon in, operator in the vector also, and as well as in the gene also, so there
Uh-huh.
could be multiple level of control
It is
on
not
the.
multiple level. It is actually, if anything, it might achieve higher stringency of IPTG induced expression. That it will operate at two different levels. One
Yeah,
in the production of T7 RNA polymerase itself, and second, T7 RNA polymerase getting access to the promoter.
right, yes,
If
yes,
you put lac operator on both the sides, then the promoter side perhaps gets occluded, meaning too crowded, so the T7 RNA polymerase cannot get recruited. That's a possibility.
yeah, yes, yes, yes.
Yeah, Akanksha.
Like, could you please explain the thing that you did in the last class, like the primer design for the like the the fusion protein and all?
फिर से
Yes, sir. Like for that part where it is getting like the fusion tag is in front of the gene.
ओके चलो करते हैं
So especially the C-terminal one,
हाँ
like uh, especially the C-terminal one, I couldn't get it.
ठीक है करेंगे
Okay,
करेंगे आयुष आपका सवाल
Yes,
क्या है
sir. Sir, I have few basic questions. One is, I wanted to know, like we are using enterokinase recognition sequence to remove
you muted yourself आयुष
needed. Hello,
you muted yourself हाँ बोलो
am I audible now?
yeah
So we have enterokinase recognition site for removing the epitopes or the tags that are unnecessary, right?
yeah
But as we can see that there is a ribosome binding site upstream to that recognition site in
yeah
the vector.
yeah
So if in case we want to remove the tag and then ribosome binding site is lost, then how is the vector?
no Ayush, you are making a fundamental mistake.
Okay.
This is the enterokinase will act on the protein, the fusion protein.
Okay,
The
not on the
tag
vector.
is there in the fusion protein only, right?
Okay.
So when that is gone, ribosome binding site is in the DNA, so there is no problem there.
Okay.
Yeah,
Okay.
Hoimi.
So, sir, my question is related to Ayush's question only. So,
Huh?
suppose I have cloned my gene, so I have a fusion tag, and my gene of interest is also there. But in between, there is no such enterokinase site. So, is it possible I can insert a site in between? I wish to like remove the tag. Is it possible, sir?
Yeah. It is absolutely possible. You simply use a vector which has the EK site and PCR amplify it, or EK site is so small that you can even design it in the primer. So let me go back to explaining the construction of fusion protein to all of you. So the like if I can manage, I will give you a little bit more elaborate description on it as well. Is it okay?
Okay, sir. Okay, sir.
Okay, so let me go back. So let me use the internet. Okay, poor Adil does not have good network. Okay. So let me do everything in front of you, okay? So sometimes net is little slow, so let us be little patient, okay? Ayush, you raised your hand again, or was
So
it?
that was my mistake.
Okay. Okay, I am putting my favorite gene. You can see my screen, right?
Yes, sir.
Yes, sir.
Yes, sir.
Thank you, madam. I'm going here, okay? You can see there are two numbers. One is NM and the other is NP. The NP is certainly the protein sequence, NM is the nucleotide sequence, okay? I'm going there. Let me familiarize you with this kind of data file. You can see, right, that I have the whole data file, okay? So, you know, like this accession number is the unique number that is specific to this particular sequence, okay? And then, you know, all the scientific papers that, referred to this thing, like that discovered this sequence, all of those are here. Nowadays, I see only the recent papers are here, not the older ones. So now here it says that 1/2/2022, Homo sapiens, mRNA, chromosome 8, map position 8q23.1, all of this is given, all the different kinds of synonyms. EIF3P46, P48, EIF3S6, INT6, these are all different. So just to give you a perspective that why this happened, okay? Like why do
So
you have
what is this 23.1 8q22? What is this 23.1?
it is map position, okay? Like any chromosome will have two arms from the centromere. On one side it is the P arm, on the other side is the Q arm. And within that, how far it is away from the centromere, that is denoted by this 23.1. I am not fully aware of it, but it's something like that. So let me tell
So
you.
these are actually the band and subband numbers on the Q arm.
Okay, very good. Who gave that answer?
Sir Ayush.
Very good, Ayush. So let me tell you the story about the name of these genes and why biologists suffer a lot because of this. So in 1980s, when scientists were trying to make a dent into cancer, that which kind of genes cause cancer, okay? So they knew that there is a virus in mouse called MMTV virus. The name itself makes it quite apparent. Mouse mammary tumor virus, MMTV virus. So they knew that if mice are infected by this virus, they develop mammary tumor. You know, it's like a kind of surrogate of breast cancer. So scientists argued that if this virus is causing cancer, therefore there is a possibility that integration of this virus in the genome is disrupting certain genes, and that's why it is causing cancer. So maybe it has certain favorite integration sites that are important for cell cycle regulation. So they took mouse mammary cell in a culture and then infected it with the virus, and tried to understand which are the cell populations that are forming tumors, and then they looked at the sequence in those and integration sites. So the integration that resulted in tumorous growth in the cell culture system, they identified the integration site, and INT is actually a like denotes exactly that integration site. So INT 1, 2, 3, 4 as a 6BN. Now, so int6 therefore was originally identified as a site where MMTB virus is integrated. That time nobody knew its relationship with protein synthesis. Later on, when we all cloned the gene, then we realized that okay, so this is a gene that is part of EIF3 subunit, etc., etc. Now the story gets even more interesting. When all these sequences became known, and in the 1990s, lots of these gene sequences was getting populated in the databases. Someone did a homology search and figured out that these genes look very similar to a famous Drosophila gene called wingless. By the way, you should know and you feel proud about it that wingless mutation was originally discovered in India, okay, in ICAR laboratory in Delhi. So wingless sequence and int sequence it was like the ortholog in human of the Drosophila wingless gene. And then many scientists explored more and figured out that many of the int genes are actually vertebrate wingless homologs. So just a minute. ক্লাস পড়াচ্ছি ওকে 10টার পরে so therefore they took the W of wingless and NT of this int series, and a new gene class was named called WNT, WNT. I do not know if you have heard of this. Wind signaling is a very, very popular signaling pathway, important for many different things, etc., etc. That was just a story. So this is how genes were originally named. Then it turned out that the same gene, some other lab discovered, they called it P48. Our lab used to call it INT6. Someone else's lab used to call it P48. So the same gene used to be named in so many different ways. So therefore, there was a very large meeting of scientists in early 2000, where they decided that all this is creating a lot of confusion. So they came to an unifying nomenclature. So now it is called eukaryotic translation initiation factor 3 subunit E, but these were all the earlier names, synonyms. Okay, so that's how it is. Okay, so now it is giving you the intron exon boundaries. So 1 to 105 is the first exon. Then the CDS is 16 to 1353, meaning at nucleotide number 16 I will get the first ATG, and at 1353 the stop codon will be there. Okay, the other exons are 106 from to 222 to 220 to 20. So this way you can also get to see how many exons were there, where is the polyadenylation sequence, and all this information you get to see. So what was the CDS? 16, right? So
Yes,
16. Okay, from here you can actually do it. Now let me see if I can copy the whole thing. I've copied the whole thing onto a Word document. Now here, this is my ATG. Someone remember it is 1335 or something like that?
1350.
Yes, sir. 1353.
So 21, 31, 41, 51. Here, you see? So it is 21, 31, 41, 51, 52, 53. Are you sure it is 1353? Let me quickly check once. 1353 it is. So 1321, 31, 41, 51. But GAA is not a.
TGA,
TGA
sir.
is a stop codon, right?
Yes, sir. Yes, sir.
So this is it. Okay. Now this is the gene sequence. Okay. Now let us get a vector sequence. So this is the PR set A sequence, okay? Where do I get the annotated sequence? You can see my internet browser again, right?
Yes, sir.
Okay. So I need someone else to open this. Okay, so I am putting it on the chat box. Correct? Someone else, please open this. Oh. So my life is certainly very difficult. Um, okay. Um, you see my Word the
Yes,
document?
sir.
Okay. So here I found the history tag sequence. You see?
Yes, sir.
Okay.
Yes, sir.
Now the right after history tag sequence, what is there? Someone can also have the presentation open. What is the feature that is right after the history tag sequence? Let us annotate it here itself.
So,
Can someone open like the slide number where this map sequence is given with
sir,
restriction
is
site
it
and
okay?
everything? Huh? It is
Is it
there
okay?
on the PowerPoint slide. If any of one of you open the PowerPoint slide,
Slide number 78. It's
whatever that.
Express.
So no, I need the codons for the express. Like after 6 CATs, what is
GAT, CTG, GAT, CTG, TAC,
hold on,
like
hold on.
that.
I am here. 79. I want you
78.
to
Yes, 79.
79. Okay. So after the histidine tag, there are some other junk sequences that are like for this purpose is not needed. So you give me the express tag sequence so that I can do a search. Can someone dictate me the express tag sequence?
It is GAT. Yes, sir.
GAT.
It is GAT,
GAT,
CTG,
GAT के बाद क्या है?
CTG,
CTG.
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 हाँ भैया कोई एक जना
Then TAC,
ये GAT CTG.
TAC,
Hold on.
TAC.
नहीं after GAT फिर से बोलो GAT. I need clear pronunciation. GAT then CTG.
CTG,
Yes, sir.
हाँ then after that
TAC,
TAC. I think we have already
GAC,
after TAC. What is it? Anyone like one of you volunteer and others remain quiet. After TAC, what is it?
GAC,
GAC, sir.
Then
Sir, the match is right, sir.
but I need the full expanse. Is
Till
that
GAT, sir. 3 more. GAT.
then
another
GAT.
Okay, so this is the total eco sequence.
Yes, sir.
Okay. So
there is an AAG as well.
The last is AAG.
AG तो है ही नहीं यहाँ पे, आप देखो स्क्रीन पे, आपको तो
Yes,
AG
sir, है
वो आगे वाला
Sir,
है sir.
it's going ahead.
GAT then AAG.
देखो GAT, CTG, then TAC, then GAC, then GAT, इसके बाद क्या है?
GAT and AAG.
AG तो है नहीं, स्क्रीन पे देखो.
सर वो आगे वाला भी एक
Sir,
GAT है ना
आगे-
सर?
Sir, आगे
third
वाला
column.
AAG. GAT, GAT, AAG.
Oh, okay, so all these will be covered as well.
Yes,
Yes,
sir.
sir.
Till GA. AAG.
Is it fine? Now the green is express tag, right?
Yes, sir.
Yes,
Okay.
sir.
Okay. So now let us look at the. After that, there are no features, right? After that, there are restriction sites. So someone please dictate to me the BAM H1 site.
GATGG,
GATGG.
GGA.
That cannot be BAM H1 site. Are you crazy? BAM H1 site has to be, it is
It was written, I
GGATCC. It's GGATCC. A restriction site has to be palindromic, right?
Yes, sir.
Okay. Let's annotate a few more restriction sites. Another restriction site, please. The next restriction site, what is there? There is BAM H1 site also. हाँ, एक एक करके सब restriction site बोलते जाओ. I will tag it here. बोलो. Next is GAGCTC.
XHO site is there.
Hold on.
CTC.
After XHO,
SAC1.
But what is the sequence? GAG.
BG.
No.
SAC1 is GAGCTC.
GAGCTC is SAC1 or XHO1?
SAC1.
SAC1 is SAC1 should be CTC GAG. Like check karo net pe.
That is 3 to 5. 3 prime to 5 prime is CTC.
But here everything is written 5 prime to 3 prime, na? XHO and SAC1 side yes. must be some overlap. Alag karke net pe check karo. I see another restriction site here, AGATCT.
XHO1 is CTC GAG.
Okay. Okay.
And SAC1 is GAGCTC.
Okay, so these two are overlapping sites. Do you see that?
Yes, sir.
Yes, sir.
These two are overlapping sites. ठीक है. Okay, so where is my ND1 site? Okay, what is the NCO1 site? CC80GG.
Yes, sir.
Yes, sir. CC80GG.
ठीक है. Okay, I think this is good enough. Okay. now see that if I don't need to take the whole thing. If I only take this part, okay, that should be good enough, right? Because I have my reading frame. Reading frame to change nahi hoga because I'll be cloning only downstream of it. Right? Is it clear?
Can you please repeat?
What I'm saying is that, can you see my Word document? What do you see now on the Word document? Do you see the gene sequence above and the vector sequence below?
No, sir.
No, sir.
No, sir. We
Okay.
cannot see a full Word document.
Okay, fine.
Yes, sir. Now we can see.
So what I am saying is that I don't need to have sequence upstream of it because my reading frame is clear from here only, right?
Yes, but sir, his tag is needed, no, sir?
But
Okay.
for the purpose of reading frame related issues, I don't need the his tag, right?
Yes, sir.
Because I know that the reading frame is continuing up to here.
Hmm.
Now let us try to clone it in here. Let us say in this SAC1 site, GAGCTC. Okay.
Sir, I did not understand this part. From where this sequence on the downside of the screen came.
I copied it from the other page and pasted it. This is the enterokinase recognition sequence.
Okay, okay.
And
So you took it from the enterokinase side. Okay.
Because the reading frame is set much before that, na?
Okay, sir.
Okay. So now let us say that this is N-terminal fusion. First, let us do N-terminal fusion. Then इसी पे हम C-terminal भी कर देंगे.
Okay, sir.
So ATG is already there. We don't need to worry about ATG. Okay, I have roughly 20 nucleotide. The good thing is that it is all small letters, very easy to identify. So we want the reading frame to be like this only, GCG, GAG, TAC, this is the reading frame, right? So
Yes, sir.
if I can only reconstitute the the BAM-H1 site, then my reading frame will be maintained because see, between BAM-H1 and this there are multiples of 3 nucleotides, right?
Yes, sir.
So this one is very straightforward, that I simply put a GGA TCC overhang and my job is done. Forward primer design is done. Reverse primer you take up to the stop codon, right?
Yes, sir.
Okay, now let us try to clone it in the NCO1 site. That for whatever reason is, I would like to clone it in the NCO1 site. Now in NCO1 site, you see here that if we go in combinations of 3, okay, so let us do it here. Fine. You can see how the NCO1 site is split now. It is in blue, right?
Yes.
Okay, so if I now put the NCO1 site here, now the restriction site will get reconstituted, right? After ligation, the restriction site will get reconstituted. The
Yes, sir.
first
Yes,
cis
sir.
was part of the last codon, then this. Now it will no longer read in your frame. It will now read as GGG as the first amino acid, right?
Sir.
So you do not want that. So what you want is that your primer will simply have one extra nucleotide. Right? If this is your primer sequence, your primer overhang is CCA TGGN, then your problem is solved.
Yes, sir.
The N can be anything, I do not remember the codon table. Like we will try to put something small, alanine or valine, something like that. Then your problem is solved, right?
Sir, I
Yes,
can
sir.
understand. Like,
Huh?
sir, after like, what is the first codon that read after the restriction digestions and ligation of that NCO1?
See, the fundamental principle is that when you digest and ligate with the same restriction enzyme,
Yes, sir.
then the restriction sequence will be reconstituted, right? So exactly
Yes, sir.
the way it is, it will look exactly like that,
Yes, sir.
right? So in that, the first C of NCO1 was part of the TSE codon.
Yes, sir.
So the codon then looked like, first C is here, then CAT, then GG. So if I did not have this N here, then this is your part of the first codon, like after ligation, how would it have looked like? After ligation, it would have looked something like this, right?
Yes, sir.
So then if this N was not there,
GGG.
then you will have a completely different reading frame.
Yes, sir.
So to avoid that, what you are doing is you are simply putting a
Extra nucleotide.
extra nucleotide there.
So primer, forward primer will contain an extra nucleotide.
Yes, power primer will contain an extra nucleotide.
Okay.
Fine?
Yes.
Okay. Now let us.
So, sir, so this problem is arising because like the NCO1 site, it is being like it is not in frame. Like when we saw BAM-H1, so it was
Yes.
distributed equally.
Yeah. That is why it is arising.
Okay, so and if we find an NCO site which is actually in frame, then we'll not need to do this modification.
Yeah. So all you need to do is when you are designing the primer, with a pen, pencil, and paper, you just do a theoretical reconstitution of the restriction site, where like if you want to really be very, very picky, then you can do something like this.
Plus, sir, another.
Sir, I have a question. This
Hold on.
NCO1 site is within this mRNA sequence. So from position 500, and this NCO1 site is within the mRNA sequence of the template that we are going to amplify. So
there is an NCO1 site within the sequence?
Yes, sir. There is an NCO site at position number 569. It is there.
Let me see. Let me see. Let me see. Control F. CCA. कहाँ पे है भाई? It is showing only
500 at 569 position it is.
अच्छा अच्छा,
there.
in the gene. In the
Yes,
gene.
sir. In the gene.
Okay, okay, okay.
Yes, sir.
So then for this particular gene, you will not be able to use NCO1 site. I did not check. I did not do all the other things that you already know to do. Okay, I did not do the restriction analysis, etc. Okay, so for whoever else was asking, you can actually do this. You can do a theoretical ligation and you can analyze the sequence after ligation. Your only consideration is that the restriction site is reconstituted and is the reading frame still maintained, right? So you see this sequence comes from the vector, ATC, TGC, AGC, like that. It comes from the vector, then the restriction site, and then it moves into the ORF. Fine?
Yes, sir.
Okay.
Sir, don't we need ATG here?
No, why should they need ATG? If you go to slide number 79,
Yes.
please go there. You see the ATG is at the beginning, right after RBS, there is an ATG.
Yes, sir.
So translation will start there, right? What extra benefit will you get by supplying another ATG for your protein, for your own ORF?
Means ATG is already there in the vector.
Yeah, that's what I'm saying.
Yes. So we have to start from this next codon, not the starting codon.
No one will murder you if you start with ATG again.
Yeah.
But it is not necessary.
Not necessary, because it will be again removed, all the methionine will be removed only after.
No, internal methionine cannot be removed. The first methionine will be removed.
Okay. So, but sir, if two methionines are there at the first amino acid and second amino acid, both are methionine, they cannot be removed at a time.
I don't think so. But in here, they will not be together, right?
Yes.
Here, the first methionine is quite far away.
Far away, yes.
उसके बाद बहुत कुछ है, उसके बाद
histidine tag, express tag, and all. Yes, yes.
If you have the ATG, no one will kill you. You can very well have it, no problem. Like in that case, the sequence will simply look like this.
Now got it, sir.
Okay. Fine. Now let me do the C-terminal cloning right here. Is that fine?
Yes, sir.
Yes.
Yes.
Okay. Let us imagine that we are now going to tag with enterokinase in the C-terminus. Okay. Okay. Okay, fine. So now you see that I have some restriction sites followed by my tag, right? These are my tags followed by some junk nucleotides, and then followed by histidine tag again. Now you want to clone your gene sequence. So what needs to happen? First of all, you cannot have your TGA. So after cloning, the sequence should read like this. So I'm first determining the reading frame. Fine. Like I have not, this is not primer sequence yet, okay? This is only, I am trying to see after ligation how the DNA sequence should look like.
So this will have ATG in the beginning though.
We are only working on the reverse primer. Forward primer पे तो यहाँ पे कोई problem ही नहीं है.
Okay.
Like you, whatever you do, it will come in this reading frame only, right? Because you are providing the ATG. If it is prokaryote, the vector will have its own ATG. Either you are going to clone in frame with that, or you are going to dictate the reading frame with your own ATG, right?
Yes, sir.
Like if you are cloning, say, in a prokaryotic vector only, which is like a PET 5A type vector, then the vector will have its own ATG. Either you will ensure that you are cloning in a manner so that the 5' end is the 5', the first ATG is the half of the NDU one side, CAT ATG wala. ठीक है ना?
Yes.
Okay. Now you want it to look like this. Now, if this is your situation, then you see there is just easy entry into MH1. So then your primer sequence can be 5' to 3'. Did I do it right?
Yes, sir.
So if you use this as the reverse primer, then after PCR amplification, you will get this as the 3' end sequence, right? Like this part. After PCR amplification, you will get this part as the like, okay, so just to make your life a little easier. So after PCR amplification, this will be the 3' end sequence, right? Of the total, like the 1300 nucleotide long double-stranded DNA. On the sense strand, the 3' end sequence will look exactly this, right?
Yes.
Yes.
After PCR amplification. So if that happens, then you will simply digest it with BAMH1, clone it with BAMH1 here, and then this sequence will automatically follow. So then after ligation, you will have this. right?
Yes, sir.
Yes, sir.
So then you are done. See, from the reading frame of the protein, it is reading into the reading frame of the tag. Eventually, it is going into the histidine tag and everything. I just eliminated that the stop codon. Any confusion? Is it fine?
Yes, sir.
Okay.
But sir, if we have another restriction site that is not
Which is
in
not
the
in frame.
yes,
Which is
sir.
not in frame. Okay.
Yes,
Let us again do with the NCO1 site only. Okay. Let's
Okay,
work
sir.
with the NCO1 site.
Also, sir, please can you put the directionality of primers as well, 5 prime and 3 prime? Then it will be clear.
This is 5 prime. This is 3 prime. Damage one site automatically dictates your 5 prime 3 prime directionality, right? Is it still being difficult to visualize?
Yes, sir.
Okay, I will do it another way. I am only designing the 3 prime primer, okay? The reverse primer. So one person, Ankit, are you there?
Yes, sir.
Okay, please read the sequence from C. I will put the reverse primer sequence here. So you understand that this is 5 prime 3 prime, right?
Yes, sir. And this is a stop codon. TG is stop
so like stop codon is not considered.
Yes.
So you start from this C.
Yeah.
बोलो.
CAT.
धीरे धीरे. C मतलब G. इसके
G,
बाद.
TA.
नहीं धीरे धीरे. C, you see me writing the nucleotide, then next one.
Okay.
C बोला आपने, मैंने किया. उसके बाद.
A, T, C, T, T.
Sorry.
Again,
Now?
C, C, G, G, A, T. So you have to write A. Yes.
Fine.
Yes.
So nobody has a confusion that this is the reverse primer sequence. And
Yes.
this is 5 prime, this is 3 prime, right?
Yes.
So now all I have to do is just put the BAMH1 side in front of it.
Okay.
But I'm done.
Yes.
Yes,
Okay,
the
sir.
only thing is we have to remove the stop codon or kuch nahi.
Yeah, और कुछ नहीं.
Yes.
Problem
Now-
arises only in the 5 prime like start codon.
नहीं, यहाँ पे भी problem हो सकता है. I'll tell you. Say for whatever reason, you have no option but to clone it in the NCO1 site. Okay. You cannot do anything other than that. Okay. For whatever reason, आपको उसी NCO1 site पे ही clone करना है. ठीक है ना?
Yes, sir.
So now your sequence must read like this. Now what happens? Just I'm breaking it into codons, nothing else. Now again the problem is that if the NCO1 site is okay, if the cloning is at NCO1 site, then none of this will be there. These are all coming from the vector. Do you understand that?
Sir, yes, sir.
So if
This is not-
NCO1 site is where we are cloning, then none of this will be there after the ligation, right?
Yes, sir.
Yes, sir.
Now you see that you are going to be off the frame. Like I'm just making it up, like I'm just trying to show you the moment I move this, you are going to be off the reading frame. Now you will not get into your enterokinase site. So you have to just restore the reading frame. Okay, how would you do that? You will simply put two nucleotides there in a manner that it codes some innocuous amino acid. Then you are fine.
Yes, sir.
That's all. Nothing else has to be done. ठीक है ना?
Sir,
बोलो.
sir, so the thumb rule is if we find a hexa cutter which is in frame, we don't have to worry about like the
Yeah.
things.
Yeah.
Okay, all right.
The moment
In
you
like
see
in like both the cases, right? N terminal or C.
the moment you see that it is not in frame, then you have to do this homework to ensure that it gets in frame by insertion of couple of one or two nucleotides. It has to be either one or two. That's it. Right?
Yes, sir.
अब सबको करना आएगा ना? This weekend again we will be testing you on this, right?
Practice once or twice and then we'll be confident.
हाँ, तो घर में practice कर लो. हाँ, you go to the NCBI site, get any random gene sequence, go to PR set sequence or any other such search for a popular vector for C-terminal tagging. Like, in fact, in my lecture slides only you have it. This PCDNA 3.1. You use that for C-terminal tagging के लिए. कर लो. And if you don't type, if you write on a piece of paper, you will be even better off than I was.
Yes, sir.
Okay. Acha, Ubaid, did Majid contact you?
Yes, sir, actually, I contacted him. I told him that you can do some efforts at the day of exam, on the day of exam. He told that he will see.
How difficult is it for him to come to your place and download the lecture once a week and go back?
Sir, it is not that far. He can come. I can also give him the pen drive. Yes, I can also go to some distance so that it will be easy for him.
Okay, so if you can help him, is it safe to travel on this road between his home and your home?
Yes, sir. Yes, sir. It is safe.
So then I would advise him that he does that, because see, IIT is sending it by post, but by the time you get to hear my lecture and discussions and he gets to hear my lecture and discussion, there is at least 7 to 10 days gap.
Yes, sir. Yes, sir.
If he collects the lecture and discussion from you once a week, then the gap will be at max 3 to 4 days.
Yes, sir. Sir, but the problem is, sir, you have to send me the lectures in the Google Drive, because I can't download them.
अच्छा. हाँ, तो Google Drive में आपको भेज दूंगा, उसमें क्या है? I have everything on my Google Drive.
Directly download. We can download it directly from YouTube as well.
हाँ YouTube में भी
Yes, sir. Yes, sir. Yes, sir. Yeah, I can download it from YouTube.
हाँ
I will download it from YouTube.
Save
हाँ
from net.
Yes, sir. Yes, sir. I will download it.
हाँ
Okay, sir. I will contact him and I will told him that I am
okay
giving you the lecture.
okay
Sir, now
हाँ
one question I want to ask in the viral expression system.
हाँ हाँ
So we talked about the three plasmid which should go into the virus: vector plasmid, packaging plasmid.
not virus, the cell.
Okay, this was the only doubt, sir. It should go in the cell.
Yeah, cell
Okay.
cell में virus बनेगा.
Okay, but that cell that virus particle will not contain the RNA genome of the virus.
No.
Okay.
So the one plasmid is where you have the gag pol env.
Yes.
That's the one that is engineered to clone your gene of interest.
Okay.
Okay. Then there is a packaging plasmid, and then there is a coat protein plasmid.
Yes.
The packaging plasmid will capture the heteronuclear mRNA from the first vector. And it will package it and it will be encapsulated by the envelope one, viral coat protein.
Okay.
Then it will infect nearby cells.
Yes.
And then it is just, like whatever was captured by the packaging proteins is now the new viral genome. Then everything will run like that.
Okay, and that will be reverse transcribed or integrated into the genome. Okay.
Yes, that will be reverse transcribed followed by integrated in the genome.
Okay. Okay.
In the first cell, I do not think there will be reverse transcription followed by no, हो भी सकता है. First cell में भी हो सकता है. Because you are providing the gag pol and env now, so pol can reverse transcribe in the first one as well.
Yeah. Okay.
Our whole lab depends on this technology only, but we are slightly lucky. The avian retrovirus, because we use avian retrovirus in chicken embryo only, so like some of the genes are endogenous to chick, so our vector is not very large. It is around 14 kilobases, and we can clone up to 1.5 kilobase size of DNA. So then we have to deal with only one plasmid. Like my entire lab works only on that technology.
Okay.
Ayush.
Sir, I have heard about this PET5A and PET5B and C. So these are artificially engineered so as to so we do not face the problem of this codon and the
reading frame.
Reading frame, right?
Yeah.
So, but I did not understand how exactly they work. So, if we could just discuss about it, might not be in the syllabus, but it would be very beneficial if you could.
See, it will be dependent on, PET5A, that is not a problem. In PR set, that is a problem because you have a fusion protein. Okay.
Okay.
So
no, I was talking about PET5B. So there is another variant of PET5A where they have one or two extra nucleotide that so that there is no
like,
reading frame problem.
PET5A, B, I have never used PET5B. PET5A particularly, let us talk about that. It gives you no option but for cloning at the NDE1 site only. There is no other cloning site. If you go to your lecture,
yes, sir.
Is अच्छा PET5A का सीक्वेंस यहाँ पे दिया हुआ नहीं है. That's why. PET5A multiple cloning site. Let me see if I can get it. वो मल्टीपल क्लोनिंग साइट नाम का कुछ चीज ही नहीं है वहां पे. Can you see it?
Yes, sir.
Unlike other vectors, here you see there is a ND1 site and then there is X bar 1 site and Basel 2 site. Okay, so you have no option but to clone it in the NDE1 site only.
Okay.
Okay, and NDE1 site comes with its own ATG, CAT, ATG. So
Yes,
as long
sir.
as you clone in that site, there is, I do not see any problem of reading frame whatsoever. That problem generally comes with fusion proteins. Let me see if I search with PET5B what I get.
Okay, sir.
Do you still see, do you see PET5B sequence?
Yes, sir.
On the screen? Okay. Let me compare with. Okay. See, the change is upstream of the ND1 side. Just look at the number, the nucleotide number in 30, like NHE1 and ND1. You see, one is 37, one is
Yes, sir.
44 in here. Here it is 36 and 43. So one nucleotide is less in here. So that must have something to do with the ribosome binding site, most likely. A ribosome binding site तो इधर है, transcription is T7 promoter किधर है? हाँ. So it must be something to do with the distance between the ribosome binding sequence and all these sequences. So I am not absolutely certain what is the utility here. I have never used this. In fact, to be honest, I have never used any of the variants. Okay, like whatever the reading frame challenge used to be, I used to typically tackle it by adding one nucleotide or two nucleotides extra in my primer. And nowadays,
there was a question. So there was a question regarding this. So they gave us a fusion protein sequence. I mean, the gene sequence for a fusion protein, and they wanted to know. I mean, they also give the sequence of PET5A, PET5B, and PET5C. So they wanted to know which of these would we select to clone the gene so that the reading frame is maintained and the protein is expressed nicely. That's why I'm asking, and I couldn't solve it. I suppose this is the topic that we are going through, so maybe we can discuss this.
This was asked for DBT-JRF?
No, not DBT-JRF. In my master's course.
Okay.
I'm from genetics background, so they usually ask this kind of questions.
Okay. I really cannot think of a utility there. And more importantly, nowadays, what we do is something very, very silly. That is, these all vectors were very useful in the 90s. Now you can design primers which are 100 nucleotide long. Okay.
Yes, sir.
So if you imagine 20 nucleotide as overlapping with the gene, then 80 nucleotide can be overhang, right?
Sir. Yes, sir.
So let me share the PowerPoint again. So if 80 nucleotide can be overhang, then if you count from here, you will be able to have an ATG followed by 6 CATs and the you don't need the leader, and the explosive epitope tag. all of this will fit in your 80 nucleotide only. So you just make a very long primer and do the PCR. You will, and here you have perfect control over reading frame and everything, and clone in any damn vector. You are fine.
Okay, sir.
Okay.
Okay. Thank you, sir.
Now nowadays life is much simpler.
Right.
Okay, fine. तो आज के लिए छुट्टी लेते हैं. I realized that the moment I declared that the syllabus will be up to this, nobody talked about mouse gene knockout at all.
Lecture is very complicated, sir. So we'll have to.
This one lecture is, so complicated. So many things.
Which lecture?
Sir, this recombinant fusion.
Complicated इसलिए है because I am emphasizing that you learn the exact nitty-gritties.
The technicalities of it.
If I only said कि ऐसे भी fusion protein बनता है, वैसे भी fusion protein बनता है, then it would not be complicated at all, and you will still not learn. Lab में जाके 4 डंडा खा के सीखना पड़ता है.
Okay, sir. We will work it out, and then if any problem, we will come back tomorrow.
So
tomorrow we will have it, or Friday?
आप बोलो, आप लोग बोलो.
Sir, tomorrow, sir. Then we can, get just like last week, we can get some time on Friday to, go through everything.
एक मिनट रुको.
Yes, sir. You can check your schedule.
मेरा कैलेंडर मोदी जी से थोड़ा ज्यादा बिजी है. हाँ तो ठीक है, कल कर लेंगे.
Okay.
कल कर लेते हैं.
Okay.
Sir, I want to ask one thing about fusion protein in mammalian cell.
हाँ.
Sir, in the plasmid, we were using 2 different promoters, one for fusion protein and another one for neomycin resistance gene. So I was thinking of a situation, if with the help of express epitome, the V5 epitome, we can find the particular, we can detect the fusion protein. So if we are able to, in given population, we are able to detect a fusion protein with this sequence. So are we using neomycin in the first stage to discriminate between 2 type of cells where the plasmid has gone or not, or we can use fusion protein directly without the neomycin detection?
No, neomycin is only for selection. Only to this plasmid, I guess, can be used to make what is referred to as a stable transformant. Okay. Let me just check one thing. Just hold on a minute. बट नियोमाइसिन के साथ उसका कोई संबंध नहीं है, फ्यूजन प्रोटीन वगैरह का कोई संबंध नहीं है. Neomycin has only to do with selection. Now the thing I am checking out is, even for transient transfection, do you need selection cassette or not? Can you are not seeing my screen, right? Just hold on a minute. See, I searched with mammalian expression vectors. Okay. You see there are 2 categories. Do you see that? For stable expression, for transient expression. Do you see that?
Yes, sir. Yes, sir.
I'm clicking on the transient expression. Just trying to see if even for transient expression, whether there will be देखो यहाँ पे नियोमाइसिन नहीं है, देख रहे हो? Do you see this does not have neomycin? Do you see that?
Yes, sir.
Ampicillin,
yes, but ampicillin is,
because every plasmid will originally be generated in very large amount always in bacteria, right? Like when you are say you received this plasmid from a company, okay? Now you will be doing a digestion, right? And you will insert your gene of interest, right? Now, where will you put the ligation mix? Directly in the mammalian cell? You cannot do that, right? So you have to first transfect bacteria with that, grow the plasmid in very large amount, and then go to mammalian cell. That's why shuttle vectors are there. Whether you are trying to express it in baculovirus or yeast or mammalian cells, shuttle vector is there because it will have to have the features for bacterial selection, propagation, etc., and your second host, whatever it is.
So here PBR322 origin is for bacterial replication,
Yes.
SV40 is for viral replication.
Yes, that will be for replication in mammalian cell.
Mammalian cell. And F1 origin, sir?
That is for bacteria. This is F plasmid. It is. I am not very sure exactly why this is needed, but this is there in every bacterial origin plasmid.
So like PRSCT also there is PUC.
Every bacterial vector will have this.
Yes. Okay.
But that is like something to do with rolling circle replication or something like that. I am not very sure.
Okay.
कभी उसके बारे में सोचने की जरूरत नहीं हुआ. Let me check some other transient thing. I should not make a case with just one thing. हाँ देखो यहाँ पे भी नहीं है.
Yes, sir.
But if I go to the stable expression vector.
सर जीन नॉकआउट में था सर.
जीन नॉकआउट में रहेगा ही, वो तो बात ही अलग है.
वो पॉजिटिव सिलेक्शन के लिए.
Vector. It is an expression vector. I'll tell you. Here you see neomycin cassette is there.
Yes, sir.
That is simply because these are non-homologous random integration of the vector sequence in the cell. To select for those where the integration has happened, you select against G418. Only those cells will survive where the plasmid sequence has been integrated in the genome.
Sir, I checked, sir. So this F1 ori is basically derived from phage, bacteriophage, and it helps in the packaging of the single-strand DNA or double-strand DNA into the virus particles.
अरे वो तो bacteriophage में, but when you are dealing with this plasmid, कोई virus की बात ही नहीं है यहाँ पे.
But it is a
yeah, that is the normal role. That is the normal rule.
Normal role, yes. But why in these vectors? Yeah.
In plasmid, like ask Google Bhaiya exactly that question. Why do you have F1 origin in PBR322?
Okay.
Ask nowadays Google is smarter than your professor. Ask directed question, you get direct answer.
No,
anything else?
For some things we never get direct answers.
Anything else?
No, sir, not for today.
Okay, I'll tell you something else, that the 17th, the next quiz dates is 17th, 18th, right?
Yes, sir.
Sir, but 17th, 18th, we have other papers,
yes,
papers.
Yes. So you will inform me in advance so that your this quiz is in respectable time gap with the other exams.
Okay, sir. Okay.
Okay, it can be any time of the day. I don't care. I don't sleep. Okay, I sleep from 2 in the morning till 7 in the morning. So besides that time zone, any time I am free. Okay. So what like I want you to be mentally prepared with is that, see, I want you to learn this technology so thoroughly so that when we move into actual topics, after this Sunday, we will move into actual topics, right? I will not quiz you on that on 17th, 18th. It is primarily because of Majid. I have decided to have a minimum 1 week gap between what I am teaching and what I will be quizzing on. Okay, so I will start teaching the next topic after this coming Sunday, but the 17th, 18th quiz will be like this. I will start with a yeast genetic screen. There will be only one question. I'll start with a yeast genetic screen, identify then identify its counterpart in yeast, in mouse or rat or human, depending on my state of mind, and then where it is expressed, then some complication which will give you a hint that if you knock it out straight, the mouse will die. So you have to do conditional knockout, or something that will give you a hint that you have to knock it out only during a certain time frame, so that you do temporarily conditional knockout. So all those you have to think through. I will try to give couple of representative examples before that, but I want you to eventually be able to think in a continuous string of all these technologies that have been taught. Once you are very comfortable with the technologies, it is much easier for me to take you through the basic topics, transcriptional regulation, translational regulation, because I will then only refer to, okay, so they did gel retardation assay to figure out the binding site. So you immediately know. I do not have to stop and explain that assay again. Okay, that is the purpose. Okay, chalo, bye, good night.
Okay, sir. Okay, good night.
Good night, sir.